Okay, so here's one for the ages. Can human cells live forever? Like if I kept you in a petri dish full of nutrient-rich media? Yeah, if you want to see more of this, let us know by hitting the like button. Also, while you're down there, don't forget to subscribe so you don't miss any of our future videos. I never let you die from anything like old age, starvation, infection, or my wrath. Would you just keep living forever? So when we think of immortal cells, we may be imagining zombies, vampires, or my brother after he spends 2 weeks without showering. But really, the first immortal cells that were ever discovered were actually human. For example, HeLa cells have divided between 40 and 60 times more than normal human cells. In fact, it's thought that the reason HeLa cells began dividing so rapidly in the first place was because they acquired a mutation that gave them this ability. This means that if you took normal cells and gave them the same mutation, they might be able to become immortal too. Some researchers think that's because HeLa cells contain extra copies of certain chromosomes. You can also select for cells that have this ability. Telomeres are sections of non-coding DNA at the ends of chromosomes. However, adult stem cells don't seem to live forever. They eventually stop dividing after about 50 divisions. But the record for the longest continuous cell division in humans goes to a skin cell that divided for 100 times before dying. Think of them like the plastic tips at the end of a shoelace. They protect important genetic information from being damaged. After all, there are no physical laws preventing it. But in reality, there are. Cells have to deal with things like the buildup of toxic substances, mutations, competition for resources, and because HeLa cells grow so quickly, they've been used to test new drugs for rare disorders that affect children. It's really helpful, since it can take years to develop a drug that works in people. But with HeLa cells, scientists can test potential drugs in a matter of days. So thank you, HeLa cells, for showing us that it's possible for cells to live forever, and changes in their environment, which means that no cell can live forever. It's dramatic. It's also scientifically accurate. No. Cell can live forever. Our cells can't live forever, except in the lab with the right mutation or genetic tweak. And in 2022, scientists created a 3D model of a tumor using HeLa cells. Well, not directly, but we did use them to grow sheep embryos. And that would have been possible if it weren't for the fact that HeLa cells can live forever. This helped them better understand how tumors grow and what causes them to become resistant to treatment. Even HeLa cells will eventually die, unless, of course, they pick up yet another helpful mutation. Every time a cell divides, its telomeres get a little shorter. Stem cells and cancer cells can live longer than normal cells, but no cell can live forever. But we're pretty good at creating conditions that allow cells to live a long time. Cancer is basically a disease of uncontrolled cell division. It makes sense that HeLa cells, which can divide really quickly, would be useful for studying cancer. But they can also be used to study things that have nothing to do with cancer. Because HeLa cells divide so quickly, scientists figured out how to stop them from dividing. When they added certain chemicals to a culture of HeLa cells, the cells stopped dividing. And when they removed the chemicals, the cells started dividing again. This led scientists to discover cyclans proteins that control the cell cycle. Cyclans regulate the timing of events that happen during cell division. And now that we understand how cyclans work, we can use them to study things like developmental disorders and cancer. And that's why we've been able to create immortal lines of human cells like HeLa cells. When they get too short, the cell can't divide anymore. But telomeres keeps adding DNA to the telomeres, effectively making them immortal. For example, HeLa cells have been used to create vaccines for polio, measles, and chickenpox. They've also been used to study cancer, develop new drugs, and even clone sheep. Wait, what? How does that make sense? How did HeLa cells help clone sheep? Just to be clear, I am absolutely not saying that we should do that. Immortal human cells have been created before, and they're used to grow new tissues for transplantation, create vaccines, and study diseases. But we'll get to that in a bit. Scientists found that most of the cells in a tumor divide at the same rate as the rest of the body's cells. But a few cells divide much more quickly. Researchers realized they could isolate these fast-dividing cells and grow them into new tumors. In fact, they found that HeLa cells were among the fastest dividing cells in the whole human body. This gives them a growth advantage over normal cells, which usually stick to dividing no more than 20 times. They come from a woman named Henrietta Lacks, who sadly passed away from cancer in 2001, but whose cells are still alive and kicking today. As we said earlier, normal human cells can only divide about 20 times before reaching the Hayflick limit. For example, there are enzymes that prevent cells from dying. So if you had lots of these enzymes, you could keep cells alive longer. That's because in 1951, a few years before she died, some of her cervical cancer cells were taken without her knowledge or consent. And then those cells, known as HeLa cells, just kept on dividing. Like even now, more than 70 years later, they are still going strong. Well, the answer is, maybe it depends on what kind of cell you are, but we will get to that. This is called the Hayflick limit. The Hayflick limit. You've heard me say it once already? The Hayflick limit. Here are some cells that can keep on dividing indefinitely. Okay, these are immortal, but they're not human. These are mouse cells. So obviously, if I wanted to prove that human cells can live forever, all I would have to do is find a way to make them divide faster than they die from other things. Keep repeating it, and soon enough it will enter your subconscious mind. Then you can use it to impress your friends. First though, let me introduce you to this very engaging video format where we zoom into your cells and watch them divide over and over and over and over again. Now let's go back to those cells. In fact, researchers estimate that in just 24 hours, a single sample of HeLa cells can multiply into over 20 million cells. And if you want to see that happen in real time, check out this video we made in collaboration with Minute Earth. It's pretty mesmerizing. Anyway, normal human cells, including the ones that make up HeLa's tumor, have only a limited number of divisions left in them, which means they have a limited lifespan. But just because normal human cells can't live forever doesn't mean that all cells can't. Well, believe it or not, scientists have done exactly that. So if you took HeLa cells out of a Petri dish and put them in a person, they could start dividing uncontrollably, leading to cancer. But you don't even need to mutate cells to get them to divide quickly. This means that it's possible to create an immortal version of normal human cells in the lab with the right mutation or genetic tweak. For now, I just want to focus on whether or not normal human cells can live forever. But there's another option too. Cells can avoid the Hayflick limit by using an enzyme called telomerase to keep their telomeres long enough to keep dividing. And guess what? Some human cells actually do this. Stem cells, which can develop into many different kinds of cells, use telomeres to stay young and keep dividing indefinitely. Theoretically, a cell could divide forever given the right conditions. That's why I said at the beginning that it depends on what kind of cell you are. And it turns out that having immortal cells has really helped advance science. Well, remember that HeLa cells are actually cancerous. HeLa cells have also been used to study viral infections, toxins, and even the effects of space travel. We've even used HeLa cells to study aging. By growing HeLa cells next to normal human cells, scientists can compare the two and learn more about what causes age-related diseases. Of course, we've also used HeLa cells to clone sheep. The discovery of HeLa cells has had a huge impact on our understanding of biology and has led to all sorts of advances in medicine and technology. I feel like we should fade out dramatically now. Started with cells dividing, and we're ending with cells dividing. Okay, thanks for watching. See you next time.